Welcome back to the Cambridge Neuroscience Interdisciplinary Seminar Series. This series features current work from neuroscientists across the schools and departments of the University of Cambridge, reflecting the pioneering work and diverse interests of our members here. Each seminar focuses on one of the new interdisciplinary themes of Cambridge Neuroscience, and for more info on the talks covered in this seminar series and all things neuro-related here at Cambridge, please follow us on Twitter at CamNeuro and follow the links below. So today, welcome to the Cambridge Neuroscience Interdisciplinary Seminar Series. Today we're delighted to welcome Professor Bill Harris. Bill was the head of department of the Department of Physiology, Development and Neuroscience, and a professor of development. I'm going to get that wrong now, Bill. I'm sure I said a professor of development. Was that your correct title?
Professor of anatomy.
Anatomy, okay. And so Bill retired a number of years, just a couple of years ago, but has since written this book in association with Princeton University Press. And I'm delighted today that I'll be able to share a link with you so that you can get a 30% discount on the book up until the 30th of November. So, Bill, I'm not going to do any further introductions because you'll be able to introduce yourself. So I'm really pleased to have you here today, and great that we have such an amazing turnout for your talk. So over to you, Bill.
Well, thank you very much, Dervila, and thank you for everyone who's listening in. Can you hear me okay? Well, okay. So I hope you can. Let me know if you can't by some kind of message.
We can hear you fine.
Okay, great. So this is a book that I wrote after I retired in 2018. And the purpose of the book was to try to engage a non-specialist audience in the field of neural development or developmental neurobiology. And it's basically a chronicle of the science that brought us to our current understanding of how the human brain is built. So I spent my career, approximately 50 years in this field, because I was trying to understand something about this, how this most complicated soft machine is built. And it's been fascinating. And during my 50 years, there have been so many breakthroughs. So Zero to Birth is organized along the same lines as this textbook here, which is called The Development of the Nervous System, which is in its 4th edition. And I wrote it with Dan Sains and Tom Ray, and now Matthias Landgraf at Cambridge has joined in. And it was really the first textbook to delineate the whole of neural development as a kind of a step-by-step process rather than a series of cool findings here and there. And it outlines the biological principles and strategies that are used in succession to build a human brain out of a fertilized egg. It's kind of like a construction guide, but there are thousands of steps involved and thousands of genes involved. And the challenge was to write a book for lay people was putting it in a way that made sense to them. And that meant dispensing with most of the jargon. So here's the table of contents. And you can see that it doesn't have a lot of jargon. The rise of the neurons, that's the first chapter. And that's about, the origin of the nervous system, where neurons come from, where brains come from. The second chapter is called the Bow Plan of the Brain, which is how the brain is organized and how it gets organized and regionalized. There's so many different parts to the brain. Chapter 3 is called Proliferation, and that's about how the population of cells that are the brain grows and grows and grows until the brain gets to be the right size. Chapter 4 is called Butterflies of the Soul, and that's after Cajal. You'll have to read the book to get the full implications of that title. But it's about how all the different types of neurons, thousands of different types of neurons, arise in the brain. Wiring up is about how connections are formed between nerve cells. Firing up is when the brain starts to become active and nerve cells begin to talk to each other. Making the cut is about the period of cell death. After many neurons are generated, lots of them die, and we'll talk about that in this talk. And then there's a period of refinement, and then the last chapter is called Being Human and Becoming You, which is specifically about how human brains are different from animal brains and how human brains are different from each other. So, oh, sorry. I have to go back one more. So from talking to colleagues in other disciplines and family members and students and supervisions and people who haven't had a university course in this field, they tend not to know much about how the brain is made. And yet I got the sense that a lot of people wanted to know something about how brains are made. And the easiest and most interesting way to do this, I thought, in the absence of too much jargon, is with stories of the main discoveries. For example, here's a story from chapter 1, The Rise of the Neurons. And here you see Hilde Mangold. She was a graduate student of a guy named Hans Spemann. And here's Hilde with her little baby son. Now, Spemann was an experimental embryologist in Freiburg in the early 1900s. And he was known as a master of microsurgery. He could do these little embryonic surgeries on tiny embryos. And what Mangold discovered was that there was a small region of a newt embryo that somehow organized the various tissues of the body around it. Work in the lab soon revealed that this organizer tissue sent out signals that neighboring cells responded to. But for 70 years, though many labs tried, no one could find any of these signals. That is until here we see Richard Harland, an ex-Cambridge PhD student, who made a breakthrough in 1992 with the discovery of a protein that was one of these signals. And he called this protein noggin because it helped make the brain. The cells just above where Richard is pointing to, these cells here, these are the cells of the organizer region. And these cells send out the noggin signal to the cells around them to become the founders of the brain, which is the first step in the construction manual of the brain. It's the creation of the founders. This is how the Adams and Eves of our brain come into being. Now, soon after Noggin's discovery, and contrary to everyone's expectations, including Harlan's, it turned out that Noggin did not promote neural development. Rather, what they found it did was it prevented non-neural development. So the cells wanted to become neural, but they were in danger of not becoming neural, but Noggin allowed them to do what they really wanted to do. It was a really exciting time in the field, which you can read about in detail in more of these discoveries in chapter 1. And it was particularly an exciting time for me, who used to teach a course in developmental neurobiology at the time and was starting to write the first edition of the textbook, to find out that finally some of the key molecular secrets of the organizer were being discovered. And now we're going to cut to the present day, also in chapter 1. As many of you know, there's been huge excitement recently as developmental biologists have learned more about how to grow stem cells in tissue culture. It's now possible to remove a few cells from any human, expose them to a regime of molecular signals so that they become like embryonic stem cells again. And we can then use signals like those that Harland and colleagues found to direct these cells to become neural stem cells, the founders of the brain. In 2011, just 11 years ago, Yoshiki Sasai of the Riken Center for Developmental Biology in Kobe, Japan, reported that if grown in the right way, these cells could form little human mini brains or brain organoids, layered neural structures, such as you see here, that are like the cerebral cortex, the human cerebral cortex, but floating around in a dish. Thanks in a large part to Tsai's work, scientists around the world recognize the huge potential of using such strategies to study human development and disease. And here in Cambridge, many labs are now using these kind of techniques to make organoids of various tissues, including brain organoids. And it's barely a decade later, and cells from patients with genetically caused neurological disorders are being used to make mini brains that float around in culture dishes that often display similar problems to those of the patient, speeding medical progress. So you can see that one of the aims of this book is to bring in examples like this that make credible links to medicine. But I also include some human stories. And again, we're still in chapter 1. This is Sasai, Yoshiki Sasai. He was a hero of mine, not just because of the mini brains, but he'd also made some of the key discoveries of the very first genes that get activated in the developing nervous system. And here he is at a huge news conference because a postdoc in his laboratory had sought instant fame by publishing a simple way to reprogram adult human cells by dipping them briefly in an acidic solution. And just as the postdoc expected, his papers made headlines. But soon other labs found they couldn't repeat his results. And an internal investigation by the Riken Center found out why they could not. The postdoc had made them up, and it was a huge scandal. And Sasai himself was cleared of having any involvement with the phony data, but he was held responsible for a failure of oversight. And he became deeply ashamed, and he committed suicide just 6 months after the publication of these papers. And that was a huge loss. So besides stories of developmental biology and links to medicine, the book also tries to expose links between the development of the brain and the evolution of the brain. And sticking with stories from chapter 1, this is a story of research that shows that written in our genes is the whole history of the brain's evolution. And that's used to reconstruct an entirely new brain in every baby. And this discovery is that much of this genetic information can be traced back to the Proterozoic eon, billions of years ago, before there were even animals. There were just single-celled creatures. Modern single-celled creatures such as Paramecia are relics of this ancient eon that preceded the origin of multicellular animals by 1 billion years. They're excitable, they're motile, they search for food, they search for mates, they adapt to new situations, they store memories of events, they make decisions. To do this 1 billion years ago, it turns out that they had already evolved versions of many of the genes and molecules that neurons use to process information. But exactly when and how neurons first evolved is still uncertain. There are evolutionary stories in each chapter, but now I'm going to skip all the way from the earliest of these eons to the most recent period of the most recent era of the most recent eon of life on Earth. And this is covered mostly in chapter 9, which is called, as you remember, Becoming Human, Being Human. What is it called? Being Human and Becoming You. In the middle of the 19th century, there was a fierce debate about the origins of human beings. Richard Owen, who is shown here on the left side of the slide, discovered the dinosaurs, and he was a famous paleontologist. And he said that humans did not evolve from monkey-like ancestors. And he pointed to the differences in the size and shapes between a human brain and a monkey brain as the strongest evidence against shared ancestry. And on the right side is Thomas Henry Huxley, known as Darwin's bulldog. And he demonstrated, however, that many of the different regions of the human brain, including many parts of the cerebral cortex, had counterparts in the brains of monkeys. Of course, nowadays, there's much more evidence that in our genes, as well as in our fossil records, that we evolved from ape-like ancestors. Compared to our closest living relatives, non-human primates, humans have expanded several regions of the neocortex, although not all areas have expanded equally. Brodmann, whose picture we see on the right here, divided the human cortex into 52 areas. Now, using modern techniques, such as high resolution, structural and functional imaging, physiology, detailed anatomy, David Van Essen at the Washington University and colleagues have now identified that the human cerebral cortex has approximately 180 different regions in each hemisphere. 160 of these also appear in the brains of macaque monkeys. To me, it's really intriguing that we seem to have about 20 regions that could not be identified in macaque monkeys. Are these new human-specific regions? If so, how did they arise? What exactly do these new regions of the brain do? Now, given that neuroscientists are still trying to figure out how any areas of the cerebral cortex, including perhaps the most well-studied area, the primary visual cortex, we still don't really know how it processes visual information in experimental animals. And to add to that, the huge challenges that arise when doing experimental work on humans, it may take a long time before we have any consensus on what these potentially human-specific brain regions are computing. So the book also points to many of these yet unsolved mysteries. The chapters between 1 and 9 cover the successive intervening steps. I'm not going to go through them all here, but I'll mention a couple that were particularly relevant to me and that helped inspire me to write the book. So here's Professor Michael Bate of the zoology department, who is a pioneer in the field of axonal navigation. And he features in chapter 5, which is called Wiring Up. Now, when I started in the field back in 1972, one of the greatest of all challenges of the field of neuroscience was to understand how the brain wires up. Michael was the first developmental neuroscientist to look at how the very earliest neurons begin to do this. Now here's a figure from the textbook. Working at the Australian University in 1976, Bates studied these cells here. These are sensory neurons at the tip of an embryonic locust leg. Here's an embryonic locust, and here's its legs, and here's one of these legs dissected. And you can see that the axons, the excitable wires that connect to the brain, are traveling from the start of these neurons along the leg, and they're heading towards the central nervous system. And along the way, they're meeting these cells, these little cells in blue here, which look like stepping stones. So they go from one cell to the next, to the next, to the next. They seem to selectively adhere to these cells. And David Bentley at UC Berkeley, who incidentally taught me my first course in neurobiology, ablated one of these stepping stone cells, and then the axons could not find their way into the central nervous system. These pioneering axons got lost. Now, let's see, I have to go back, sorry. I'm gonna ruin this joke here. So the pioneers are effectively adhering to the stepping stones, and axons of the later arising neurons selectively stick to the pioneers and use them to get into the CNS. So Michael Bate used to do a set of lectures for part 2, final year undergraduates in neuroscience at Cambridge University. And when I took over from him these lectures, he lent me his slides. And one of the jokes that he used to engage students on the course was to ask students how they thought the brain wired up. And then he showed them this, which I mistakenly already showed you in advance. Now, I use this ploy in the book, though I changed Kevin's name here to the more gender neutral Terry. And I wrote, imagine a tiny Terry, the electrician, hiding inside an unborn baby's brain, consulting a color-coded wiring diagram and plugging cables into the right sockets. But you know that's not how it works. Neurons manage to wire themselves up without Terry's interventions. Bate was extremely encouraging to me in writing this book. He was a pioneer himself and a mentor for me and many neuroscientists, including this person, Corey Goodman, who made huge advances in understanding how the brain wires up. Goodman was a student of David Bentley, the guy who ablated that stepping stone cell. And Goodman showed how later arising neurons could use the trails that the pioneering axons had blazed. Here, drawn by me, an example of one of the very few images in the book, are the growing tips of 2 axons. This one we'll call A, and this one we'll call P, in a locust embryo that are studied by Corey Goodman when he was doing his groundbreaking work at UC Berkeley in the 1980s and 1990s. So you can see axon A and its growing tip is selectively adhering to this previously grown axon, which is a pioneer. And this axon called P is selectively adhering to this one, and it's growing posteriorly, and this one's growing anteriorly, which is why this one's called A, and this one is called P. You can really see this selective, specific stickiness. These growing parts could reach all these different axons, but they selectively adhere just to some and not others. And Goodman began to search for the molecules that were responsible for this differential stickiness between axons. And he visited Michael Bate at Cambridge University then, and together they began to do the same sorts of experiments on embryos of the genetically tractable fruit fly, Drosophila, as we call it in the field. And fruit fly mutants, Drosophila mutants, provide access to the genes involved. Goodman and colleagues were about to reveal the molecular nature of axon guidance and wiring in the brain, which, as I said, was a complete mystery when I started in the field. And it was a tremendously exciting time for developmental neurobiology. It turns out that many of the genes that the Goodman lab found were cell adhesion molecules. And one of the property that most of these cell adhesion molecules share is that they are homophilic, which means that they love themselves. They bind to the same cell adhesion molecule on the surface of another cell, and the 2 cells stick together because they have these cell adhesion molecules on their surface that love each other because they're the same. In the developing embryo, if 2 axons express the same homophilic cell adhesion molecule on their surfaces, they tend to adhere to each other and they make a bundle or a fascicle of axons. So the first 2 proteins that the Goodman's lab found in this way were called fasciclins. Fasciculin 1 was found on axon fascicles that cross the midline, while fasciculin 2 was found on axon fascicles that are running from head to tail. And follower axons like these that you see here, A and P, use these pathways. If they want to cross the midline, they express fasciculin 1, and if they then wish to grow in a head to tail direction, they switch to fasciculin 2. So the developing nervous system, as it turns out, uses homophilic molecules and CAMs like a color coding system. Red axons stick to red axons, yellow axons stick to yellow axons, and so forth. And the idea that emerged from Goodman's work is that many axons travel to their destinations in a way that is a little like how Bostonians travel to the zoo. They take the green line to Park Street, they get off there, switch onto the red line, getting off at Ashmont. From Ashmont, they take the number 22 bus to Franklin, and they walk the rest of the way themselves. I use a lot of such metaphors in the book to avoid using jargon. Now, chapter 5 ends with a short bit about axon damage and the search for ways to help severed axons regrow in adult humans. Research in this field has revealed that adult mammalian neurons are intrinsically less able to regrow than our young neurons. They appear to have lost the mojo of their youth. Research also indicates that the site that bears the scar of the neural damage is often full of materials and molecules that inhibit the growth of axons. And another huge challenge is the fact that many of the guidance cues that were used by axons to find their targets during initial outgrowth in the embryo are no longer available to guide regrowing axons in the adult, and the distances are much greater. So while there's been some serious progress in understanding the underlying challenges for regeneration, there's still a huge amount to figure out if we are going to be able to cure paralysis. Wiring up is indeed one of the great feats of the developing brain and continues to be one of the great challenges, greatest of challenges for neuroscience and medicine. Okay, here's a little story from chapter 7, which is called Making the Cut. And it's a metaphor really from a set of ice hockey videos that I watched. You know, I love ice hockey. And this was a reality series where a bunch of amateur players tried out to make professional teams with professional coaches, and only the best made it. And I'm going to skip chapter 6, which we've skipped, which is about making synapses. But once synapses are made, neurons begin to communicate with one another. And this begins a whole new period of brain development, which is in chapter 7, Making the Cut. Up until this point, building the brain has been a task of construction. But once neurons are established, the balance shifts toward demolition. Young neurons compete to make effective synapses. Indeed, making good synapses is a life or death matter for billions of your neurons. The overall chances of survival for a young neuron is only about 50%. Why would you build the brain this way? Why not make the right number of neurons in the first place? In the book, I suggest that it's a little like how a hockey coach might build a team from those who try out for different positions. Only the best make the team. But of course, like there's no Terry to wire up the brain, there's no coach involved in the selection process here. The neurons select themselves for the brain team by fighting with each other. And those that receive and make good synaptic connections survive, while the others don't. And here you're looking at Victor Hamburger. He was a graduate student of Hans Schmemann. Remember, he started off in chapter 1. Victor Hamburger was in love with Hildy Mangold. That story's not in the book, but I found out later. Anyways, he was doing his postdoctoral studies in Chicago when the Nazis came to power. And being a Jew, he stayed in the USA and ended up at Washington University in St. Louis. And here is Rita Levi-Montalcini. She has a parallel story. Levi-Montalcini was forced out of her job at the University of Turin in Italy and went into hiding with her Jewish family. And it was here that she read Hamburger's paper. And she was so excited that while she was in hiding, she set up a laboratory in her room with the most basic equipment, where she repeated Hamburger's experiments. And those experiments of Hamburger involved microsurgery, Speymann's influence, on microscopic chick embryos in the egg within just a few days after they were laid. He would remove the limb bud. Here you can see a fine tool that's removing this tiny limb bud from this 2-day-old embryo. And a few days, and then a week or so later, he would look at the motor neurons by cross-sectioning the spinal cord, and these red cells are supposed to be the motor neurons. Motor neurons are the neurons in the spinal cord that innervate the muscles. And what he found was that there were fewer neurons on the side that where the limb was ablated. So Hamburger, probably under the influence of Speymann still, imagined that the limb bud induced the production of motor neurons. But Levi-Montalcini in her hideout in Turin got exactly the same results as Hamburger, but with a totally different explanation. And that's because she looked at the spinal cords of the embryos on successive days following the removal of the limb. And what she saw was that most of the motor neurons had already been generated on both sides, but that as a consequence of the operation, motor neurons began to die on the side without the limb. Now, after the war, Hamburger invited Levi-Montalcini to his lab, and they worked closely together for many years. Perhaps their most remarkable finding was that even in normal development, the spinal cord makes about twice as many motor neurons as survive. And the same is true with frogs, chicks, mice, humans. They discovered that the synaptic partners provide survival factors. And Rita Levi-Montalcini went on to receive the Nobel Prize for her work on the isolation of the first of these neuronal survival factors, known as nerve growth factor. We now know that there's an exceptional potential for such neuronal survival factors, of which there are now known many, for the treatment of a multitude of neurodegenerative disorders, as well as brain and spinal cord injuries. Medical research on neurotrophic factors is very active, and a huge amount of progress has been made on our understanding of how they work. However, translating this research into effective therapy for humans has been challenging, in a large part due to the blood-brain barrier, which prevents proteins and peptides like nerve growth factor that can be injected into the bloodstream to ever get to the brain cells that need them. But once a young neuron has made the cut and established enough synapses, it now has a good chance of surviving for a lifetime. Chapter 7 is full of other life and death stories, which you might like to read. But the last story I'll tell here comes from the ending of the book, and it concerns the work of Marion Diamond, shown here at UC Berkeley. She died 5 years ago. A lot of Berkeley, it turns out, in this talk. Here she's explaining something about how the human brain, something about the human brain to a youngster. Now, Marion Diamond was particularly fascinated by the question of how early postnatal experience affects the microscopic structure of the brain. And she was first attracted to this question by a story that Donald Hebb told her. Hebb is one of the heroes of chapter 8. I'm not doing chapter 8 in this talk. Anyway, the story about this was that he brought home a pair of freshly weaned rats as pets for his 2 children. And his children adored the rats, and the rats liked the kids. And the rats had the run of the house, and the kids played with them all the time. And Hebb thought that these lucky rats have had the benefit of an enriched and interesting life compared to their poor siblings that he had kept back at the lab, who still lived in cages. So he decided to challenge his children's rats against the lab-reared rats in a maze running competition. And the lab-reared rats turned out to be no match at all for the pet rats. So when Marion Diamond heard this story, she decided to repeat that story in a more controlled way. And she made enriched cages with lots of rats and toys in them versus isolated cages with just one rat in it and no toys. And then she looked at the brains of animals that had been raised in either deprived or enriched conditions. And she found that in enriched rats, the cortex was generally thicker, and in deprived rats, the cortex was generally thinner. And the underlying mechanisms involved in deprivation involved Hebbian-like strategies of synaptic weakening and elimination, while those of enrichment involve strategies of synaptic strengthening and maintenance. You might expect a book like this to take a stand on the issue of nature versus nurture in shaping our brains and identities, and it does. As I say in the introduction, and as I emphasize using many examples throughout the book, our evolutionarily inherited genomes, that is our nature, hold the basic building plans of the brain. And our interaction with the environment, including the intrauterine environment, nurture, affects and guides the building process. Conversely, the influence of the environment is often affected by the genetic differences among individuals. So nature affects nurture, nurture affects nature. And in building a brain, you can't ignore another major player, lady luck or chance, which also has a significant role in generating brain developmental variability. Because of differences in genetic makeup between everyone, differences in the in utero environment, and differences due to the variability, randomizing influences of the way biology works, everyone's brain differs from everyone else's from the moment of birth. And my feeling is that more than 90% of the building of brain is done by this time. And these differences between individuals only grow as the final shapings of our brain is done in the outside the womb and incorporates our individual experiences of the womb. So one of the lessons I learned from writing this book is that what makes us all human makes us all different. And though the brains and personalities of identical twins tend to be more alike than those of non-identical twins, the most different thing about identical twins is their brains and their personalities. Now, when I was an undergraduate at Berkeley, this is a funny little story, not in the book, I remember going into a bookstore to buy a book for my sister's kids, and that was Fox and Socks. And I also bought this book, Sexual Selection and Animal Genitalia. And the cashier thought it was a very strange selection of books for one person to buy. Now, the thesis of the second book, The Sexual Selection and Animal Genitalia, is that genitalia have evolved to be quite different in different animal species and can be very useful in telling similar species apart. But I'm going to venture here to speculate that the most different things about closely related species of animals is also their brains. So now I've told you a few of the stories in the book, and some of you, I hope, might want to read the whole thing for the full experience. I want to thank Princeton University Press and the various friends and family members who suffered through drafts of this book, to Cambridge Neuroscience for hosting this talk, and you for your attention, and I'm happy to take any questions at this point.
Thank you, Bill, for that wonderful overview and a real insight into your new book. All of your colleagues here at Cambridge wish you the very best with it. Join us next time when we welcome Professor Alan Herbison, Professor of Neuroendocrinology in the Department of Physiology, Development and Neuroscience, and supported by the Wellcome Trust. Alan is interested in all things hypothalamus and fertility, and he'll be speaking to us on hypothalamic episode generators underlying the neural control of fertility. Don't miss it. For more info on the talks covered in this seminar series and all things neuro-related here in Cambridge, please follow us on Twitter @CamNeuro and follow the links below. See you next time.